Okay, so we move on to the next talk. Dr. Sanjiv will talk about cardiac regeneration, hope and heights.
Thank you. Can you guys hear me?
Yeah, you're loud and clear.
Thank you, Dr. Said, for the kind introduction. And can you see the screen as well?
Yeah, it's visible.
Thank you. So, good evening, everybody in India. Uh, I would like to begin by thanking the organizers, Dr. Kartha. Dr. Kartha, good to see you. And Dr. Harikrishnan, Dr. Jayakumar, and all the other members of the organizing committee. It's a little early here in Winnipeg for me, but I must tell you guys that my excitement is not of attending this meeting is not less than any of you who are sitting in India. So really glad to be the part of this exciting meeting today. I have no disclosures to make related to this presentation. Now, I don't need to tell it to this audience that heart failure is turning out to be number one killer in today's world. But stem cell therapy has shown lots of promise and hopes in terms of cardiac repair and regeneration. Several animal studies suggested that after transplantation in the heart, stem cells are able to improve cardiac function, and based on that outcome, there have been lots of clinical trials in different phases conducted in cardiac patients. So the point that I want to make here by telling all this is that there have been lots of promise and lots of hopes around stem cell therapy being turning into treatments for heart failure. But the title I was given by the organizers for this meeting was Hopes and Hypes. And I would say, yeah, there have been some hype as well in terms of cardiac regeneration when it comes to stem cell therapy. Uh, Many reports even suggested that heart is a self-renewing organ. It can regenerate itself through endogenous mechanisms. And there have been several other reports which made big claims by saying that exogenous stem cell therapy, transplanted stem cells can differentiate into beating cardiomyocytes and integrate with the host myocardial tissue and help in reviving the cardiac function. Uh, so that all being said, the outstanding point here is that uh, is there it is there a chance of hope above all these hypes? I mean, I would say if we are able to overcome the challenges associated with stem cell therapy in terms of clinical translation, yes, then we can start saying that there are some hopes in terms of cell therapy turning into clinical treatments. And one of those challenges that has been noted by experts in the field is survival of implanted cells in the heart. And I'm going to talk about uh, some of these challenges, and we'll also talk about some strategies that are being taken to improve survival of implanted cells in the heart. And when it comes to cardiac repair using stem cells, different types of cells are being considered or tested in different stages. And out of all these, bone marrow derived mesenchymal stem cells have taken the lead because these cells are easy to isolate. There are no ethical issues involved with this cell type because the source is always adult tissue. Uh, they secrete several growth factors and angiogenic factors, and these cells are considered to be immunoprivileged because These cells do not express cell surface immune antigen, major histocompatibility complex class 2, and they secrete several immunosuppressive soluble factors and are able to suppress the proliferation of cytotoxic T cells and induce production of Treg cells. These are the mechanisms of MSC's mediated immune suppression when you transplant them to any host tissue. So, due to these properties, bone marrow MSCs in different preclinical studies in several small as well as large animal models, bone marrow MSCs of autologous or recipient derived cells were able to improve cardiac function. And based on the positive outcome of these animal studies, there have been several clinical trials conducted with autologous bone marrow MSCs in the last 15 to 20 years, I would say. The outcome of these trials was positive, but it wasn't as effective as the outcome of animal studies was. And different reasons were given to explain muted response of clinical trials, but age-related limitation or age-related decrease in the regenerative or proliferative ability of autologous cells was the major issue, because in animal studies, though they were mostly conducted in young recipients, whereas patients enrolled in clinical trials were in their advanced age, because that's when cardiac disease takes place. So it was thought that allogeneic bone marrow MSCs from young and healthy donors are probably the ideal cell type for cardiac repair. Again, in animal studies, when you transplant allogenic MSCs, they are able to improve cardiac function and promote angiogenesis in the infarct areas. And the outcome of initial clinical trials with allogenic bone marrow MSCs was again promising, positive, but the long-term fate of implanted cells in these patients, and I would say even in animal studies, wasn't assessed. So we became interested in understanding the fate of implanted cells after implantation. What we did was we implanted both autologous and allogenic MSCs in a rat model of myocardial infarction. As we can see here, after 1 week of cell implantation, both autologous and allogenic MSCs were able to improve cardiac function. After 3 months, cardiac function started deteriorating in allogenic cell recipients. After 5 months, as you can note here, there was no difference found in the cardiac function in allogenic cell recipients and in control group. When we assessed the survival of implanted cells in the heart, after 1 week, both allogenic and autologous cells were present in the heart. After 5 weeks, almost all the allogenic cells were rejected by host immune system. In a separate study, we found that there were antidonor alloantibodies specifically against implanted cells in the heart. And later on, the outcome of our studies was confirmed by several other reports. Just want to highlight a very good paper that was published in 2014 in Nature Biotechnology. I'm going to read some of the sentences as it is from this paper. It says that immunogenicity of bone marrow-derived MSCs needs to be recognized as a characteristic of these cells, and its impact on stem cell therapy needs to be examined. So there is a need for in-depth analysis of immune properties of bone marrow MSCs if you want to take these cells to the clinic. With this, one of the areas of research in my program is to understand the immunological behavior of bone marrow MSCs under ischemic and hypoxic conditions. We also study effect of cell differentiation on their immunogenicity. Under this theme, we focus on IPSCs or induced pluripotent stem cells and their derivatives. The immunoprivilege of bone marrow MSCs is preserved by several immunosuppressive soluble factors which are secreted by these cells. Uh, Two of my PhD students who graduated last year, Dr. Sareen and Dr. Abu Al-Rab, they got interested in this work. What they did was they screened several soluble factors in MSCs before and after exposing them to hypoxic conditions. What they found was that prostaglandin E2 or PGE2 levels decreased in hypoxic cells. PGE2, as we know, is an immunosuppressive soluble factor. It promotes immune suppression by decreasing the proliferation of cytotoxic T cells, natural killer cells, B cells. It also inhibits maturation of dendritic cells. The question that we wanted to address at this stage was this decrease in prostaglandin E2 that we found in hypoxic cells. Is it associated with loss of immunoprivilege or increase in immunogenicity? And to find this out, we did several experiments. We co-cultured stem cells from V-star rats with lymphocytes from Lewis rats and studied the effect of lymphocytes on stem cells in terms of cytotoxicity. As you can see here, the level of cytotoxicity increased towards hypoxic cells, and treatment with PGA2 decreased it. So one of the properties that MSCs have is they are able to suppress proliferation of lymphocytes, as I said in the previous slides. So we also assessed the level of lymphocyte proliferation in our co-culture studies. As you can see here, normoxic MSCs were able to suppress lymphocyte proliferation, and they lost this ability after exposure to hypoxia. You treat the cells with PGE2, they are able to suppress lymphocyte proliferation. So PGE2 preserves immunoprivilege of allogenic MSCs. In hypoxic MSCs, the level goes down, cells lose their immunoprivilege. If we maintain PGE2, they are able to suppress lymphocyte proliferation. So in the next set of experiments, we wanted to test whether maintaining PGE2 levels in transplanted cells would preserve their immunoprivilege and prevent their rejection in vivo conditions. But the challenge for us was to achieve a sustained release of PGE2 in the heart, because PGE2 has a very short biological life under physiological conditions. So what we did was we synthesized a biodegradable temperature-sensitive hydrogel and used this hydrogel to deliver prostaglandin E2 along with stem cells to the heart. And this hydrogel facilitated sustained and slow release of PG2 in the heart. For our in vivo setup, we used stem cells from V-star rats and transplanted around 3 million cells with or without hydrogel to the infarct area in a rat model of myocardial infarction. After 5 weeks of cell implantation, we assessed the survival of cells and host immune response. In both the control groups, MSC and hydrogel, almost all the cells were gone. But in PG2 group, there was a significant increase in the number of cells that we found. host immune response in terms of anti-donor alloantibodies came down when Pg2 was supplemented through a hydrogel. So Pg2 again preserves immunoprivilege of allogenic MSCs under both in vitro and in vivo conditions, and it prevents rejection of implanted cells in the heart. I couldn't show the data here, but we also saw some improvement in cardiac function. Uh, as I mentioned, we also are interested in understanding the immunogenicity of induced pluripotent stem cell derived cardiomyocytes. This is particularly important because iPSCs or iPSC derived cardiomyocytes are now being investigated in clinical trials. And the first clinical trial in cardiac patients is just concluded by Dr. Yoshiki Sawa from Osaka University. Just a little bit of background. iPSCs, these days you can reprogram any somatic cell in our body to turn them into, to induce pluripotency, to turn them into stem cells. And these stem cells can be differentiated into any cell type in the body. And the concept is that patient-matched pluripotent stem cell lines can be created and they can be used as personalized medicine for disease modeling and for cell therapy. When it comes to cardiac repair using iPSCs or iPSC-derived cardiomyocytes in different animal models, iPSC-derived myocytes were able to integrate with, it is already reported, were able to integrate with the myocardium and improve cardiac function. So this is on a positive note, but there have been several challenges reported with this cell type as well. And in 2011, I think this paper came in Nature, and they reported that iPSCs or iPSC-derived cardiomyocytes, even if they are from autologous source, they become immunogenic after differentiation. So with this, we wanted to investigate the immunogenicity of iPSC-derived cardiomyocytes and mechanisms of it. Two of the lab members, Glenn Lester, who's finishing PhD this year, and a postdoc, Dr. Srivastava, they are focusing in this area. What they do is they would take blood cells from healthy individuals, humans, and then they would generate iPSC colonies from these blood cells and differentiate iPSC towards cardiomyocytes. What we have found so far is that when you differentiate iPSCs to cardiomyocytes, the levels of MHC2 or HLA in case of human cells, HLA-DRB and HLA-DQB increases. We are now taking proteomics or RNA-seq approach to understand the mechanisms. We have found that these iPSC-derived cardiomyocytes, they become immunogenic compared to iPSC stem cells. I'm not going to go into details of this because of time limitations, but in the next 1 or 2 minutes, I just want to talk about some of the strategies that
Sir, could you conclude because we won't have time for discussion, please?
Just 1 minute, sir.
Okay.
So some of the strategies that we are taking to improve the survival of implanted cells in the heart. And a team of engineers, Dr. Rafirad and Wang Yan, who are working on this project, what they have done is they have developed mexine, or which is a carbon-based material, mexine quantum dots. What we found was that these quantum dots are immunosuppressive in nature, and they are highly biocompatible with mesenchymal stem cells, and iPSCs or iPSC-derived cardiomyocytes. And in terms of mechanisms, we found that these quantum dots, mixing quantum dots, are able to suppress the proliferation of cytotoxic T cells and promote induction of T regulatory cells. We are now developing platform in terms of chitosan-based hydrogel by incorporating these quantum dots in the hydrogel to deliver stem cells to the infarcted heart. I'll stop here, but just want to mention that biomaterials and stem cell-based approaches may have future, but we still need to find right combination of cells, scaffolds, and factors responsible for myocardium repair. And we need to understand basic biology behind all this, and extensive collaborations among clinicians, scientists, and engineers is going to play a vital role in all this. So I want to end by saying that when there is a promise of hope, there is always a danger of hype. The onus is on us how honestly we report our data, how honestly we put our efforts in it. I think together we can achieve it. So I'll stop here. I want to thank the lab members who contributed to the work I have shown today. I want to thank funding agencies, especially Canadian Institute of Health Research, Research Manitoba. Major portion of funding came from these two sources, and my collaborators in different parts of the world. And I'll take any questions if you guys have. Thank you. Sorry, I think I went a little over.
Thank you, Dr. Sanjiv. I think you've shown us very clearly that whenever we talk of any research, when you move from bench to bedside, you really have to go through the steps, because I think for stem cell therapy, we move probably a bit too fast from bench to bedside. We move towards the bedside. I think the clinicians were really desperate for a therapy for heart failure because we were losing the patients. So we pulled the therapy to bedside. We started injecting patients with stem cells. Initial results were hopeful, but then we realized that initial hope was probably too much to hope for. And we really need a lot of bench work before we can actually take stem cell therapy as a given. I think are there any questions from the panel? Okay, if not, we can have, any questions?
I see something typed here. I don't know what that is.
That's a congratulations, I think. We'll have some time at the end. If there are any questions, we can take them up later also. So we'll move now to Dr. Ajay on chemotherapy-induced heart failure. Dr. Ajay, please.
Thank you.